Hello everyone, welcome back. This is NPTEL Immunology Week 2. This course is coordinated by Professor Ghosh and Professor Ganguly from the Department of Bioscience and Biotechnology at IIT Kharagpur, and I am Bias Mukherjee, a research scholar from the Department of Bioscience and Biotechnology, IIT Kharagpur, and my supervisor is Professor Day from Department of Bioscience and Biotechnology. And I am re-recording this class because this particular class recording, there was an issue with the recording, so I'm re-recording this. And let's begin with today's class. Okay, so in last class we had talked about the immune responses. So we had talked about innate immune response and adaptive immune response in details, and we had talked about the immune system, the cells in the immune system, the tissues in the immune system, and so on, right? And the organs of the immune system, etc. So in today's class we are going to talk in details about inflammation, about immune responses, adaptive and innate immune responses, the different proteins that are involved in this immune responses, the cytokines, chemokines, and so on. And we are going to talk about B cell and T cell maturation in a lot of details. And we are going to talk about the structure of antibodies, the different classes of antibodies, and related topics. Okay, so let's begin with today's class. So we'll start with the immune system. Immune response is a well-coordinated defense system. The immune response is a relay between innate and adaptive immune response that we have talked about in the previous class. This is just to give you an idea and understanding of what the innate immune response and adaptive immune response is, in case you guys have forgotten. Okay. Now, if the threat persists, so initially what starts off is the innate immune response. As soon as any pathogen enters into the body, the innate immune response begins, and then it gradually uh, calls the adaptive immune response into play if the pathogen is a new pathogen, or it is there in a large number, or something is so devastating that the innate immune response cannot deal with it alone. Okay. Now, innate immunity, of course, is the first line of defense, and there are four different barriers that include anatomic barrier, physiological barrier, phagocytotic, and inflammatory response. Okay, the key cells of the innate immune response are neutrophils, macrophages, dendritic cells, mast cells, etc. There is no memory formation, that is, the response intensity is the same as repeat exposure. Okay, so no memory is formed, but this is a very fast response and it is very generic, it is not specific. Now coming to adaptive immunity is highly specific. That is why it takes time to get started with. Okay, it takes around a week or so to get started with, but this is very, very potent, and the memory is retained for years, even for a lifetime, if that's a particular kind of pathogen that comes in. Okay, and the two branches are cell mediated and humoral immunity. So in humoral immunity, antibodies are involved, and in cell mediated immunity, the T cell and the B cells are involved. Okay, now we will move on to the layers of defense of the innate immune response. So there is the anatomic barrier, and there are the psychological, physiological barriers, right? So the anatomic barriers include skin and mucous membrane, which are the physical barriers that physically prevent the microbes from entering into a system. Then we have the physiological barriers. This includes body temperature rise, that's seen in case of fever. Then we have the low pH in our stomach of pH 2, which kills the microbes that we eat along with our food, and we drink the food and the drinks. Okay, and then secreted enzymes, so lysozyme in our tears, saliva in our mouth, and mucous membranes, mucus secretion. So these have lysozymes, and these create conditions in which pathogens cannot survive. Then we have the phagocytotic barriers. So there are cells in the immune system that engulf the pathogen, they eat up the pathogen, and that constitute the phagocytotic barriers. Then we have the inflammatory response. So in inflammatory response, a lot of different cells are called into place. For example, neutrophils, dendritic cells, etc. What happens is there is swelling, there is redness, there is pain, and there is increased temperature in that area. And this is a link between the innate immune response and the adaptive immune response, because a lot of different cells are pulled into that place. A lot of cytokines are released, and then what happens is maturation of B and T lymphocytes, activation and maturation is also triggered, and adaptive immune response kicks in. Okay. So we have talked about all of these in the last class, and that is why we'll just brush through these topics quickly, and then we will start off with today's main topics. Okay. Now, here is everything in details once again, the anatomic and physiological barriers. So skin, mucous membrane, secreted enzymes, stomach acidity, fever, and commensal microbes. Now in last class I was telling you that there are microbes on our body, inside our body, and they are either benefiting us or they are not causing us any harm. So the ones that do not cause us any harm, they are called commensals. Okay, and these commensals are actually preventing other pathogens from coming and attacking us. For example, Staphylococcus epidermidis is naturally present on our skin, and it prevents Staphylococcus aureus to come and sit on our skin, because Staph aureus causes pimples on your skin. Okay, it causes these pimples and blisters and all that. Now we will move on to the immune cells. So all of these immune cells that are involved in phagocytosis and inflammation, they are majorly originating from the myeloid progenitor cells. Okay, and the B cell and T cell and the associated cells involved in adaptive immunity, they are coming from the lymphoid progenitor cells. So the two groups are getting formed from the hematopoietic stem cell, which also self renews. Okay, so the cellular first responders are our neutrophils. Okay, there are the macrophages, there are dendritic cells and mast cells. So neutrophils are majorly active during inflammation. So they are fast acting immune cells. They first migrate to the site of infection in large numbers. Macrophages, most important work of the macrophage is to phagocytose and present antigen. So the tissue resident um, phagocytes, these are macrophages, they engulf pathogen and activate gene expression upon internalization. Okay, then we come to the dendritic cells. Now, dendritic cell is a link between the innate immune system and the adaptive immune system. Okay, now these dendritic cells, they are antigen presenting cell. They also phagocytose and then they present antigen, and they travel to the lymph nodes and they alert the T cells. Okay, so next we have the mast cells. Now mast cells, they have a lot of small granules inside of them, and what happens is during this time of need, these granules break open, and from that granule a lot of chemokines and cytokines, these are released. For example, histamine is released from these granules. This drives vasodilation, so the blood vessel is getting dilated and a lot more blood flow is allowed, increasing vascular permeability. Okay, now we will look at phagocytosis in details once again. So if you remember from our previous class, there was this pathogen which was coming in, it was attaching to the cell membrane of the macrophage. Then macrophage was engulfing it, so the membrane was going around it and engulfing it into a vesicle, and this vesicle is called a phagosome. Now the phagosome was going and fusing with the lysosome present inside the macrophage, and it was forming a phagolysosome. So all the contents of the lysosome are after all degradative enzymes, right? Proteolytic enzymes. So these enzymes are now degrading this entire pathogen. So what is happening? The pathogen is now getting broken up into smaller bits and pieces, and these smaller bits and pieces are getting presented in the form of antigen, on the surface of the macrophage or surface of the dendritic cell or whatever it is, okay, whatever this phagocyte cell is, and it is getting expressed on the surface using a special type of protein called major histocompatibility complex. Okay, it can be MHC class 1 or it can be MHC class 2 depending upon what type of pathogen it was or what type of antigen it was. Now, if it was an indigenous antigen, that is, it was either from a viral protein that was produced inside the cell, because our cell using its own machinery produced that viral protein, then it is going to be expressed using a MHC class 1, or it can be from a virus infected cell, or it can be from a diseased cell, okay, and the pathogen is endogenous pathogen, endogenous antigen. So then it is presented using MHC class 1. The other one is MHC class 2 presented antigen, which happens when this pathogen comes from outside. So it is exogenous. Okay. Now, monocytes, neutrophils, tissue dendritic cells, and tissue macrophages perform phagocytosis. After phagocytosis, the activated macrophage or dendritic cell goes to the lymph node to activate B and T cells, right? So now it is getting connected to the adaptive immune system. So what happens is, following this, after this activation, humoral and cell mediated immunity kicks in. After phagocytosis, cytokines are released to attract more immune cells to the site of the wound. Okay. Now we will look at what happens in phagocytosis and how is it that a cell distinguishes whether a particular protein or antigen or whatever it is a foreign being and not something from our own body. Okay, so if you remember last class, we had talked about PAMPs and DAMPs. Now, what are these PAMPs? PAMPs are pathogen associated molecular patterns and DAMPs are disease associated molecular patterns. So, PAMPs are what is recognized by the pattern recognition receptors in our body on the surface of this bacteria or any glycoproteins that are present on the bacteria. Okay, these are PAMPs. What about DAMPs? DAMP is the signal that is given by a dying cell. So let's say there is a virus infected cell and or there is a tumor cell, and on its surface a lot of proteins will be expressed, and these pattern recognition receptors will identify those DAMPs and they will understand that these are actually something that needs to be killed, something that needs to be phagocytosed. Okay, so the next step is engulfment or phagocytosis. Then what happens? The phagosome fuses with the lysosome and forms the phagolysosome. Then what happens is it is degraded into small peptides, bits and pieces, and then it is displayed in the form of antigen display on the surface using either MHC class 1 or MHC class 2. Then antigen presentation happens. So this dendritic cell or this macrophage now travels to the lymph node presenting this antigen on its surface, so that either TH cells or cytotoxic T lymphocytes, they can identify these antigens presented on the surface and they act upon it. Okay. So this is how antigen display happens. Okay. So the antigen is taken in case of MHC class 2 antigen presentation, and in case of MHC class 1 antigen presentation, it is a virus infected cell or a tumor cell, and then what happens is uh, it is presented. So here is an MCQ. Now in the class, all of you had answered all of these MCQs, but here since we do not have any students, I'll be showing you the answer. Okay. The question is, a macrophage recognizes a bacterial cell wall component via a pattern recognition receptor before phagocytosis begins. Which receptor family is primarily responsible for this initial PAMP recognition? Option A is FC receptors, option B is toll-like receptors, option C is complement receptors, and D is MHC class 1 molecules. So the right answer would be toll-like receptors, and the explanation is that toll-like receptors or TLRs on macrophages and dendritic cells recognize conserved PAMPs such as bacterial lipopolysaccharide or flagellin. Flagellin is the protein that is present on the flagella of the bacteria, which is the motility appendage of the bacteria, right? And this recognition precedes and triggers engulfment. So makes it engulf this, right? FC receptors bind antibody coated targets. Complement receptors bind complement opsonized target. MHC class 1 is used for antigen display, not initial recognition. Okay. Acha. Now this opsonization and what is an FC receptor, etc. We will talk about this in the upcoming slides of today's class. We talked about opsonization in last class also, if you remember, but we are going to talk about this in more details in today's class. Okay, let's move forward. Now, all the cells, they release, all of these immune cells, they have a property to release a lot of chemicals to ask other cells to come to this site of the wound. And these chemicals are cytokines. Okay, they are of multiple different classes and they have different functions. Now we will look at a few of them now. So signals from an activated macrophage. So once a macrophage internalizes a pathogen, gene expression is triggered and cytokines are released. These immune systems primary cell to cell messenger. Okay. Now, chemokines such as interleukin 8 attracts more neutrophils and immune cells to the site of infection. Interleukin 6, it induces hepatocytes or liver cells to secrete complement proteins. Now, what is complement protein? Complement system refers to a group of proteins, which is a huge group, and there are like 9 proteins or so, but then these proteins, they undergo cleavage and they break into 2 different fragments, and these 2 fragments have 2 different roles. Okay, so in this way all of these proteins break into fragments and they form a massive cascade of reactions. So one after another the cleavage happens and activation happens, and each of these proteins have some definite roles and they interact with something and they perform a lot of work. Okay, so we will talk about complement system today in brief, but majorly we will talk about the complement system in our upcoming classes of this particular course. Okay, the next thing is raising of body temperature. Okay, and that is fever, so that the pathogen dies, right, slowing the pathogen replication. Then we have interleukin 12. Now interleukin 12 and related cytokines bridge the innate response into adaptive immune response. How? Because interleukin 12 promotes the production of TH cells, so T helper cells. Okay, right. So we will see whether it is TH1 or TH2 cells. We will talk about that in the upcoming slides. Right. So now inflammation. Remember from last class we had studied about the events of inflammation, but today we are going to look at it in a lot more details. Okay, so we remembered that there was this tissue damage, there was this wound that had happened, bacteria had entered into the body. Then what happened was tissue damage causes release of vasoactive and chemotactic factors. What is chemotactic? Chemotactic factors are once again factors that are asking other immune cells to come to this place. Okay, that trigger a local increase in blood flow and capillary permeability. Permeable capillaries allow an influx of fluid exudate and cells. So exudate is the fluid part without the cells, and cell is the cellular part, right? Okay. So now what happens is phagocytes migrate to the site of inflammation through chemotaxis, and phagocytes and antibacterial exudate destroy the bacteria. Okay. Now see what is happening. The neutrophil it undergoes a lot of different um, things, right? First thing is it rolls and it attaches to the endothelium of the blood vessel, and then it squeezes between these endothelial cells and it passes out and goes to the site of wound. So exudate contains complement system proteins, antibody, C-reactive protein. So it is containing the fluid part of the blood along with all of these important proteins and chemicals, okay, but it is not having the cells as such. Okay, so now this we had seen in the previous class, but in today's class we are going to see exactly how these neutrophils travel to this site of the wound. Okay, so neutrophil extravasation, there are 4 steps in this. The first step is after infection the circulating neutrophil numbers can rise up to 10-fold. So 10-fold means, let's say initially they were 100, then they will become 1000. Okay, so that is 10-fold increase. They cross high endothelial venules into tissues in 4 sequential steps guided by cell adhesion molecules. Now, cell adhesion molecules are molecules that are present on the surface of these immune cells as well as endothelial cells. So, basically all cells, right? So, the first step is rolling. What happens in rolling? There is a particular cell adhesion molecule called mucin on the surface of this neutrophil. So, this mucin is going to interact with another cell adhesion molecule called selectin, which is there on the surface of the endothelial cell. So these two are going to bind. Then what happens? This neutrophil, see what happens is it has receptors for interleukin 8, and interleukin 8 is going to be released, and interleukin receptors are there on the neutrophil, so it comes and binds to it and it triggers integrin conformational change. Now, this integrin is present in the form of a cross. And what happens once this um, interleukin binds to the interleukin receptor, this cross-shaped integrin becomes this parallel integrin molecule. And this parallel integrin molecule is now able to bind to this ICAM, which is there on the endothelial cell. This results in adhesion or attachment. So these are all sequential steps, right? And finally what happens is transendothelial migration, that is the neutrophil squeezes through the endothelium and then through diapedesis. Diapedesis means it is moving like an amoeba and it is going into the tissue towards the pathogen. So we will look at all of this in the form of a figure so that you can remember better. Okay. See, there are 4 steps. The first one is rolling. Second one is activation. The third one is arrest or adhesion. The fourth one is transendothelial migration. So what is happening? The first step, there is interaction between mucin, which is there on the neutrophil, and selectin, which is there on the epithelial cell. Now E-selectin is because this is on epithelium. If it would be L-selectin, it would be present on leukocytes such as lymphocytes and neutrophils. Okay, it can also be P-selectin, that means it is present on the platelet. So now for now we are looking at E-selectin here because it is there on the endothelial cell, right? Now see what happens. So the step 1 is this interaction. Step 2 is interleukin goes and binds to the interleukin receptor on the neutrophil, right? Next what happens on the neutrophil, there was this cross-shaped integrin protein. This cross-shaped protein becomes this parallel protein and this integrin is now able to bind to this Ig superfamily ICAM, and then it adheres or binds to this endothelial cell. Then what happens in the fourth step, it squeezes between 2 endothelial cells and leaves for the wound, okay, site of the wound. So that is what is happening. Now we have chemical mediators of inflammation or effector molecules. For example, histamine. Now histamine is there in the granules of the mast cells, and when these are released, it causes vasodilation, increased vascular permeability, opening HEBs for cell migration, and this is blocked by antihistamine drugs, right? Anti-allergens. Now, histamine is very, very potent in allergies, and we'll be talking about allergies in our upcoming lectures, okay? Then we have prostaglandins. So, from the mast cell and macrophages, prostaglandins are released. They also increase permeability and contribute to pain and fever. So, aspirin, and then this anti anti-fever medications, right? So these are all anti-pain medications. These are all helping with this prostaglandin. So they are basically blocked by NSAIDs. Now these NSAIDs are non-steroid group of drugs, and they are helping with uh, prevention of prostaglandin activity. Then we have leukotrienes. Leukotrienes are also released from mast cells and macrophages. They increase the vascular permeability and smooth muscle contraction. Then we have anaphylotoxins. Now, complement cleavage products. So let's say there is this protein called C3. This cleaves into C3A and C3B. Now, both of these have two different functions. Okay, C3B has a different function. C3A has a different function. So, now this C3A is involved mostly in inflammation. So these are chemoattractants that trigger mast cell degranulation. Degranulation means that the granules are all breaking apart and histamines are getting released. Okay, amplifying histamine release. That is what is happening. Now we have two MCQs. Now I must say that all the people who had joined today, you had answered very well. But unfortunately, right now, since this is being re-recorded, you all are not there. So let's go through the MCQs, okay? During neutrophil extravasation, firm adhesion to the endothelium immediately prior to transmigration is mediated by which pair of molecules? And the options are selectins on neutrophil, mucin on endothelium, integrins on neutrophils, ICAMs on endothelium, chemokines on neutrophils, and TLRs on endothelium, cadherins on neutrophils, and selectins on endothelium. Okay, for the second MCQ, it is which inflammatory mediator synthesis pathway is specifically blocked by NSAIDs such as aspirin. So, the options are histamine, leukotriene, prostaglandins, and anaphylotoxins. So the answers that we have are during neutrophil extravasation, integrins on neutrophil and ICAM on endothelium, they bind at the adhesion step before transmigration of the neutrophils. And for the second one, we have NSAIDs inhibit prostaglandins by working on the cyclooxygenase enzyme that converts arachidonic acid into prostaglandins. Now, arachidonic acid is a 20 carbon fatty acid chain. Okay, it's a long chain fatty acid, 20 carbon fatty acid, and this is getting converted into prostaglandin, and in between this cyclooxygenase is the enzyme that helps in the conversion, but then NSAID blocks it. Now we will talk about all of this in details, okay, eventually in this course, but not today. So let's move on. So now we will talk about the two branches of adaptive immunity. Both branches originate from the lymphoid precursor in the backbone, in the bone marrow, and are triggered when the antigen presenting cells deliver processed antigen via MHC molecules. So the two groups are cell mediated immunity and humoral immunity. Now cell mediated immunity is driven by the T lymphocytes. So cytotoxic T lymphocytes, helper T lymphocytes, and a wide variety of subsets of this helper T lymphocytes are involved in this one. Coming to humoral immunity, antibodies are involved in humoral immunity. So it also requires help from the T cells, and that is how the two systems are basically linked. Okay. Now we come to cell mediated immunity. This is driven by T lymphocytes They mature in the thymus, and that is why the name T lymphocyte comes. The naive T cells become CD4 containing helper T cells or CD8 containing cytotoxic T cells. So what is CD? CD is cluster of differentiation. This is a group of proteins that are showing similar immunological activity. Okay. So that is cluster of differentiation, and helper T cells have CD4 receptor. Cytotoxic T lymphocytes have CD8 receptors. CD8 receptor containing cytotoxic T cells directly kill infected target cells. These can be virus infected cells, these can be tumor cells or diseased cells, and they are recognizing the antigens that are presented by MHC class 1 antibody. Okay, CD4 cells are helper T cells, and they have a wide variety of subsets: TH1, TH2, TH17, Treg, and TFH. So TFH is follicular T lymphocyte, T helper T lymphocyte. This coordinate the activation of B cells. Okay, and so all of these are basically helping out at different stages. Now we have humoral immunity. This is driven by B lymphocytes. They mature in the bone marrow. They are activated in the lymph node germinal center. Antibody mediated, the B cells differentiate into the plasma cells and secrete antibodies or memory B cells. And they require help from the T cell. So follicular T helper cells drive activation and affinity maturation and class switching. Once again, affinity maturation and class switching we are going to talk about in our upcoming lectures. So coming to cell-mediated and humoral immunity, if you remember, in cell-mediated immunity, there was this pathogen which was presented by MHC class 2, and this was recognized by the Th cell, and the Th cell got activated. In turn, it went, and what happened is you have your T cell receptors recognize antigen bound to MHC molecules. Binding antigen MHC activates T cells. Then activated Th cells secrete cytokines that contribute to the activation of B cells and cytotoxic T lymphocytes and other immune cells. So activated cytotoxic T lymphocytes recognize and kill the altered self cells, and they also recognize the MHC class 1 presented antigens, right? Now coming to the humoral immunity. Now B cells do not require any antigen presenting cells to recognize the antigen. They can directly bind to the antigen. That is one of the major differences between B cell receptor and T cell receptor, that your B cell receptor directly binds to the antigen. And what happens is it internalizes the antigen, so it interacts with the antigen and differentiates into antibody secreting plasma cells, and antibody binds to the antigen and facilitates its clearance from the body. Okay, that is the entire story of cell mediated and humoral immune response. Okay, so now we go to the T cell receptor and co-receptor. So in T cell receptor and co-receptor, if you remember from previous class, that there was this virus infected cell which was presenting its antigen using the MHC class 1 receptor, and this was getting identified by cytotoxic T lymphocyte. See here, besides the TCR, that is the T cell receptor, there is another receptor. This is called the co-receptor, and this is CD8 in case of cytotoxic T lymphocyte. This is helping to identify this MHC class 1 molecule, and it is helping to interact with this antigen. Okay. Then we have the Th cell. Th cell recognizes the antigens that are presented by MHC class 2, and once again your CD4 receptor is helping, CD4 co-receptor is helping in this identification and interaction. Okay. Now we come to T cell maturation in thymus. So this is a very important topic. So please read properly. Pay attention for now. So T cell maturation in the thymus. T cell precursors leave the bone marrow. enter the thymus and are selected in stages by the thymic epithelial cells by expressing MHC class 1 or class 2. So initially they are double negative, that is there is neither CD4 co-receptor nor CD8 co-receptor there. Initially, no co-receptors expressed yet in the T cell precursor. T cell receptor gene rearrangement occurs across the DN1 to DN4 stages. So DN is double negative. Okay. Now it has both the co-receptors. So it becomes double positive. So TCR is now expressed along with both the co-receptors. The cell is not yet committed to a lineage. Now what does that mean? Commitment to a lineage means whether this cell is going to form the cytotoxic T lymphocyte or it is going to form the helper T cell. It hasn't decided yet. So it hasn't committed to any lineage yet. Now the next one is selection by thymic epithelial cells. So here what happens? There are two types of selection. The first one is called positive selection and the second one is called negative selection. So in case of positive selection, what happens is if there is MHC1, it should interact, and then what happens is CD8, the fate is selected. Okay. If there is MHC2, again it should interact, and as CD4, the fate is sealed. But if there is MHC1 and MHC2 and it does not interact with any one of these, then what happens is the cell dies from neglect and there is apoptosis. Okay, because it is showing affinity for both of these types. It is not showing affinity for any of these types of receptor. Okay. Now, the next one is negative selection. So what happens is in negative selection is that there is an MHC and there is a self antigen. So a protein from your own body. It is there. And let's say that this T cell goes and binds to it very tightly. Then what happens? It is a threat for our own body. And it is killed. And this process is called central tolerance. And it is killed by apoptosis. Okay. Now this positive selection happens in the cortex, negative selection happens in the medulla. Okay. Lastly, what happens is single positive, that is either CD4 or CD8 cells are selected and naive T cells exit the thymus and travel to the lymph node awaiting antigen to get activated. Okay. Now these are all, you can think them in the form of exams of the cells, okay, of the T cells, of the B cells, and if they clear the exam, they are alive. If they are not clearing the exam, they are dying from apoptosis. Okay, so it is shown in the form of a figure here. Okay, you can see that this cytotoxic, this common lymphoid progenitor is there, and from that common lymphoid progenitor, it is going through a lot of different stages, and inside the cortex it is forming double positive. Early DP is double positive. Then there is if it is still double positive, then it is killed by positive thymic selection in the cortex. Then what happens is if it is interacting with the self antigen, it is killed using the negative thymic selection inside the medulla. Okay, and that is happening. Now we have an MCQ. During the T cell development in the thymus, a double positive thymocyte that shows no productive interaction with the self MHC on thymic epithelial cell will, option A, differentiate into CD4 helper cells, option B, differentiate into CD8 cytotoxic cell, option C is undergo death by neglect, that is apoptosis, and D is become a regulatory T cell. So the answer would be C, that is undergo a death by neglect, apoptosis. So the explanation is that positive selection requires a productive T cell receptor interaction with MHC, with self MHC, class 1 or class 2. Thymocytes that fail to engage the self-MHC at all receive no survival signal and die by apoptosis. That is death by neglect. Only those appropriate MHC1 or MHC2 engagement are positively selected towards the CD4 plus or CD8 plus lineage respectively. Now, the same thing is written here in a lot more details. So, you can go through this, okay, whenever you get time. Now, we will talk about T cell activations and the two signals that are required for T cell activation. Now, this single positive T cell with its lineage fixed, it comes to the lymph node, okay. In the lymph node, it meets these antigen presenting cells presenting the antigen using MHC class 1 or class 2. Now, besides this TCR interaction, there is also interactions with the co-receptor, plus there is another interaction with another co-receptor. Okay, so total 3 interactions happen. The first 2 interactions result in activation of the T cell. The third interaction basically helps in maturation of the T cell. Okay, so the first interaction is helper T cell activation. So MHC class 2 and T cell receptor. So remember, T cell receptor is CD3. Okay, cluster of differentiation 3. And CD4 co-receptor for exogenous antigen. They are all interacting. Cytotoxic T cell activation, that is MHC class 1, T cell receptor and CD8. The antigen is obviously viral or self antigen. These results in activation of the T cell. Okay. Signal 2 or co-stimulation. So there is another interaction between another receptor called CD28 on the T cell binds to B7. Now this B7 is CD80 or CD87 on the antigen presenting cell. Without this second signal, the T cell does not fully activate and may become anergic. What is anergic? Once again, it is killed by apoptosis. It is becoming completely non-functional and it is becoming dormant and it is killed by apoptosis, right? So result, T cell maturation happens. The T cell expresses the IL-2 receptor and secretes IL-2 receptor. So T cell produces IL-2 and it has receptor for IL-2. So it constantly, there is this production of IL-2 and which is helping its own proliferation and differentiation into effector and memory T cells. Okay. Now we have a small MCQ regarding this. A T cell receives signal 1, that is T cell receptor and MHC engagement, but no CD28B7 co-stimulation. The most likely outcome is option A, full activation and IL-2 secretion. Option B is differentiation into memory T cell. Option C is energy, that is functional unresponsiveness. And option D is immediate apoptosis via pass ligand. Okay. So, the right answer in this case would be option C, that is energy. Full T cell activation requires 2 signals. One is antigen recognition, that is signal 1, and the second one is co-stimulation via CD28B7, CD28B7, that is signal 2. Antigen recognition without co-stimulation typically renders the T cell anergic, unresponsive to subsequent stimulation rather than activating or killing it outright. Okay. Now we have the TH helper subsets, TH1 and TH2. How are they forming? Okay, so after this TH cell has decided its lineage, it undergoes activation, it undergoes maturation. Then what happens? Depending upon the cytokines in the environment, it can either become an TH1 cell or it can become a TH2 cell. Okay. Now to become a TH1 cell, it requires a lot of interleukin 12 and it itself produces a lot of interferon gamma and TNF beta. Okay. And these are again having specific functions such as delayed type hypersensitivity reaction, macrophage activation, opsonization, complement fixing immunoglobulins, etc. Okay. The second one is Th2 cells, which are formed when there is a lot of IL-4 in the environment, and it itself also produces IL-4 and IL-5, and it causes eosinophil activation. IgE and some other classes, antibody classes, are produced in majority. Right, so this is basically helping in B cell maturation and antibody formation. So we will now see this in writing. Okay, so T helper cell subsets Th1 and Th2. The cytokine environment during activation determines which CD4 plus subset a helper T cell becomes, each with a distinct downstream function. So Th1 is formed due to a lot of IL-12, and it secretes interferon gamma, interferon alpha, and it activates macrophages to kill the internalized intracellular pathogen, drive cell-mediated immune response against viruses and intracellular bacteria. Okay. Then we have the Th2 cell. So this Th2 cell formation is driven by interleukin 4. It secretes these and drives the B cell class switching to IgG, activates eosinophils and basophils. The key is anti-parasite immunity, response, and allergy. Okay, so the main role is here. Okay. Now we go to the cytotoxic T lymphocyte. So this cytotoxic T lymphocyte has CD8 co-receptor, and it directly interacts with the your infected virus infected cell or tumor cell, and it kills it. Okay, so using three main effector molecules. The first one is called perforin. Perforin is a pore former. Okay, so this molecule goes and forms pores in the cell membrane. So it forms pores in the target cell membrane, allowing entry of other effector molecules. Then there are granzymes. So these granzymes are actually present in the form of inactive proteins inside so they are inactive proteases, okay, that are present inside the cytotoxic T lymphocyte. So now remember, enzymes are present in the form of zymogens, right? So zymogens are inactive enzymes. So that means that when this is present in the form of granzyme inside the cytotoxic T lymphocyte, of course it is not degrading the cytotoxic T lymphocyte itself because it is inactive. It can undergo activation through various mechanisms such as cleavage. There can be proteolytic digestion, there can be any kind of post-translational modification. So there are different ways in which a zymogen becomes an active enzyme. That is happening here too. Okay, as soon as it is entering into that cell through the pores created by this perforin, it is becoming active, and now it degrades all the protein and induces apoptosis. Then another machinery is fast ligand and fast receptor mediated activity. So what happens is there is this fast ligand on the surface of this cytotoxic T lymphocyte, and this fast ligand binds to the fast receptor that is present on the target cell, and it directly triggers apoptotic pathway. So the result is the target cell undergoes programmed cell death or apoptosis, and the pathogen's replicative niche is eliminated. So cytotoxic T cells are killing the target cell when there is interaction between the TCR and the CD8 receptors. Then what happens? The granules are breaking open, granzymes are getting released. Granzymes, as soon as they enter into the target cell, they become activated. Perforins are forming pores through which the granzymes can pass into the cytotoxic T lymphocytes, and then the entire cell is degraded through apoptosis. Plus there is also this fast ligand on the surface of the cytotoxic T lymphocyte that is interacting with the fast receptor, and it is mediating apoptosis once again. Okay. Now we have two more MCQs. Which cytokine combination is most characteristic of a Th1 polarizing environment, and what is its principal downstream effect? So the options are IL-4 or IL-5 eosinophil activation, interferon gamma and TNF alpha macrophage activation, interleukin 17 and interleukin 22 epithelial chemokine induction, and the fourth option is interleukin 10 and TGF beta and suppression of effector T cells. Okay, so granzymes delivered into a target cell via perforin exert their cytotoxic effect primarily by A is forming membrane pores directly, activating caspase dependent apoptosis through protease activity, cross-linking fast receptors, degrading viral nucleic acids. Okay, so the answers are B, that is interferon gamma, and TNF alpha, that is tumor necrosis factor alpha. So Th1 cells are induced in an IFN gamma interleukin 12 rich environment and themselves secrete interferon gamma and TNF alpha, which activate the macrophages to kill intracellular pathogens. IL-4 or IL-5 defines the Th2, that is eosinophil IgE responses. IL-17 or IL-22 defines the Th17. So these are other Th cells. Okay, so you can read about them if you want. Then we have granzymes. Obviously, perforins form pores through which the granzymes entered. And once inside, the granzymes are serine proteases and they activate the caspase cascade, driving the target cell into apoptosis. Also, fast ligand and fast receptor are also mediating apoptosis, right? Now we have B cell activation in the germinal center. So that was all about T cell activation. Now we are going to talk about B cell activation. So inside this lymph node, there are regions called germinal centers, and there the B cells are sitting waiting to get activated. Okay, so let's see what happens. These helper T cells are the ones who are basically helping the B cells to get activated. Okay, now an IgM bearing naive B cell binds antigen through the B cell receptor. Now remember, B cells can directly interact with the antigen. That is the characteristic property of the B cell, and that is what distinguishes it from the T cell, right? That B cell receptor directly interacts with the antigen. It does not require any antigen presenting cell to mediate this interaction. Okay, now B cell processes antigen and expresses it on MHC class 2. The B cell interacts with the helper T cell through three additional required contacts. One is TCR MHC2 and CD4 interaction. The second one is CD28 and B7 interaction. The third one is CD40 and CD40L interaction. These are once again co-receptor interactions, right? Now we have antigen cross-linking of the B cell receptor delivers the final activating signal. And then together these four interactions license the B cell to enter the germinal center and begin the dark zone light zone reaction. So we are going to look into this in details now. Okay, so entire thing that we read about, we are going to look at that in this figure. So see what happens. Initially there is this naive B cell. Okay, this is your naive B cell, and the naive B cell is either directly interacting with the antigen or it is interacting with this Th2 cell presented antigen. Okay, it is presenting using this CD40L and CD40 interaction. There is this T cell receptor interaction along with MHC class 2. So all these interactions are happening, and because of that, what is happening is that this T follicular helper cell is helping this B cell to undergo proliferation. Then the B cell can uptake two different pathways. It can either go for a germinal center independent pathway, where it can produce memory B cells and plasma cells very, very quickly. But then these plasma cells are short lived and they are not very specific, and the antibody quality is also not very great. Or it can enter into the germinal center. Now, germinal center has two zones. One is called the dark zone, the other one is called the light zone. So, in the dark zone, it is called dark zone because a lot of cells are there, and under the microscope, it looks dark. Okay, so a lot of cells are there with a lot of large nuclei and very small space for cytoplasm because the cells are constantly dividing. The B cell here undergoes clonal expansion and BCR diversification. So what is clonal expansion and BCR diversification? It means that the same antibody, it can bind to, let's say, only one antigen, but then let's say that this particular pathogen has multiple antigen, 5 antigens, and we want this antibody to be twitched in very small ways so that it can very specifically bind to all these 5 antigens. So how does that happen? This region of this antibody, the binding region, FAB region, this undergoes a lot of genetic mutations. Those are called somatic mutations, and that is enabling a large number of different varieties of antibodies to get formed so that they can bind to these different antigens with very high affinity each and very high specificity each, right? Coming to the light zone. Light zone is light because here the cell division is relatively less. Fewer number of cells are there. They are spaced out. They are not appearing dark under microscope. They are appearing light under microscope, and there is spaces also in between. So here what happens? Affinity maturation happens. So once this specificity has been changed, it comes out into the light zone. Now we want to see how tightly it is binding to this antigen. And the ones that are binding tightly are selected. That is your affinity maturation. Then what happens? Then once again your long-lived plasma cells are produced, and they are giving very high quality, highly specific antibodies, and there are the memory B cells. Okay, so this is all about the germinal center dependent memory B cells, and the previous one was germinal center independent memory B cells. Okay, so that was all about B cell maturation. So now we will look into the dark and light zones once again. So the germinal centers can be either dark zone or light zone. In dark zone, what happens is rapid proliferation. Activated B cells, now called centroblast, divide rapidly. Somatic hypermutation is happening. That means the antibody variable region genes, they accumulate point mutations, generating a diverse pool of B cell clones. And obviously it appears dark under the microscope. That is why it is called dark zone. That is due to dense population of cells with large nuclei and less cytoplasm. Okay, coming to the light zone. Now in the light zone, affinity selection happens. Follicular dendritic cells select the B cells, now called centrocytes, producing high affinity antibodies. Low affinity cells die by apoptosis. Class switching happens, and the selected antibody switch class, then differentiate into plasma cells or memory B cells. The outcome is that plasma cells, they secrete antibody, and memory B cells, they are mostly high affinity IgGs. Okay, these are formed. Now, antibody have certain effector functions. Okay, so how do antibodies basically function? Now, antibodies alone cannot destroy a pathogen. So, they recruit other effector systems to finish the job. Now, antibodies act in three main ways at the site of the infection. The first one is neutralization. In neutralization, what happens is, if you remember from the previous class, that there was this toxin, antibody was coming and coating the entire toxin, making it neutral, right? So, antibody binds antigen directly, blocking toxin activity or pathogen entry into the host cell. The next one is opsonization. This is even more powerful. What happens is, let's say there is this bacteria and the antibody comes and coats the entire pathogen, and the entire pathogen is kind of now easily engulfed by the uh, phagocytotic cells like macrophages and neutrophils, and it is eaten up. Okay. Then once again antigen presentation and activation of the adaptive immune response and everything else happens. The other one is complement activation. So what is complement activation? Antibody binding triggers the complement cascade. Okay. So antibody mediated complement activation is another way. Now complement system, we will look into it now. So there are three different ways the complement system gets activated. One is through antibody binding, that is called the classical pathway. Then there is the alternative pathway, and the third one is lectin binding pathway. So now we have two MCQs here in the germinal center. Somatic hypermutation of immunoglobulin variable region genes occur predominantly in which zone and is followed by which selection process? So the options are light zone, selection by T cells; dark zone, affinity based selection by follicular dendritic cell in the light zone. Then we have dark zone, negative selection by macrophage. Okay, so somatic hypermutation happens in the dark zone, and affinity based selection by follicular dendritic cell in the light zone is what happens. Now, which set of interaction is required for cognate T cell helper to a B cell during germinal center activation? So you have TCR MHC, CD8B7 Fas and Fas ligand interaction. You have TCR MHC2, CD4 and CD28B7, CD40, CD40L interaction. Okay. So remember B cell along with T cell is CD40, CD40L. Okay. Right, so let us look at the right answers. So the right answers are B for the first one and once again B for the second one. So explanation is that centroblasts proliferate and undergo somatic hypermutation in the dark zone. The resulting centrocytes migrate to the light zone where the follicular dendritic cells present antigen and select cells producing higher affinity antibody. Lower affinity clones are eliminated by apoptosis. Okay, and for the second one, you know that the second one is the right answer, right? Okay. The complement system. The complement system is a group of proteins which are produced by liver cells. Okay, they are mainly produced by liver, circulating in an inactive state. Okay, and they are 9 proteins. So C1 to C9, they are 9 proteins mainly, and then they undergo a cleavage for activation. So each and every one, they undergo cleavage. They break up into two, and these two have two separate functions. Let's say C3b breaks up into C3ba. Sorry, so C3 breaks up into C3a and C3b. Okay, and the three pathways are there. One is classical, one is antibody triggered, and the other one is so classical or antibody mediated, the other one is alternative, and the third one is lectin mediated. All of these converge on C3. Okay, C3 undergoes cleavage, it forms C3a and C3b. C3b is mainly responsible for opsonization. So C3b goes and coats the pathogen surface, tagging it for phagocytosis. Okay, now C3b, C5b, C6, C7, C8, C9, all of these together come and form a big tunnel. Okay, they're forming this hollow tunnel, and this hollow tunnel, let's say this is the cell and pathogen infected cell, it goes and sits on this cell and it ruptures this and it causes cell lysis. So this is called membrane attack complex. Okay, this entire tunnel is called membrane attack complex. Now C3a and C5a, they act as anaphylotoxins. Now we will talk about anaphylotoxis, anaphylaxis and all this in the upcoming classes. So what happens is this C3A and C5A, they are mainly involved in recruiting leukocytes to the site of inflammation. Okay, so this is how it looks like. So there is a C3 cleavage and C3b, C5b, C6, C7, C8, C9, all of them form MAC, that is membrane attack complex. C3b is involved in opsonization and C3a is involved in inflammation. Okay, that is the function. And complement system is a group of proteins of nearly 30 plasma proteins, mainly made by the liver, circulating in inactive form, that get sequentially activated in a proteolytic cascade. Each protein cleaves or activates the next to help clear pathogen. It is a part of innate immunity but also links to adaptive immunity. Three activation pathways converge to a common endpoint. Okay, classical pathway is triggered by antigen antibody complexes, for example, C1q binding antibody. Lectin pathway is triggered by mannose binding protein called lectin binding to microbial sugars. Okay, and you have alternate pathway triggered spontaneously directly on microbial surfaces. So all of the three converge to cleave C3, and opsonization, that is C3b, coats the pathogens, tagging them for phagocytosis. C3a and C5a are anaphylotoxins that recruit and activate immune cells. Membrane attack complex C5b to 9 punches hole in the pathogen membrane, causing lysis, mainly effective against gram-negative bacteria. Right. Now we come to the basic structure of an antibody. So a typical immunoglobulin is Y-shaped molecule with two identical heavy chains and two identical light chains linked together by a disulfide bond. Okay. Now antibody is a very important topic, obviously. So expect questions from here, and also this is not a difficult part. So it's a scoring topic. So make sure that you learn very well about antibodies. Okay. Now, a typical immunoglobulin is a Y-shaped molecule with two identical heavy chains and two identical light chains linked together by a disulfide bond. Now, the variable region, that is variable region in heavy chain and light chain both, the N-terminal domains that determine the antigen binding specificity, highly variable between the antibodies, so the immune system can recognize diverse antigens. Okay, these are the FAB regions, or the antigen binding fragments. Then we have the constant regions. They are there in the heavy chain as well as light chain. In the next slide, we have a diagram for this antibody. Now see, they are having conserved domains similar in structure across antibodies of the same isotype. So what is an isotype? IgA is a different isotype. IgA, G is a different isotype. IgD, IgM, all of these are different isotypes. Okay. Then we come to hinge region, a flexible segment between CH1 and CH2 that lets two antigen binding arms to move independently. That is called a hinge region. FC region, CH2 and CH3 mediates effector functions such as complement activation and binding to FC receptors on immune cells. So these are the effector functions of this FC region. Okay. Now let's see what happens to this antibody. So this is the entire structure of an antibody, entire diagrammatic representation. So you can see that there are these FAB regions. These are the FAB regions, and there is this heavy chain and light chain. Heavy chain is of 5 different types, light chain is of 2 different types, and then you have the antigen binding region and you have the effector region. Okay, the constant region is majorly active in the effector activities. Okay, and there are a lot of disulfide bridges, multiple disulfide bridges in this antibody. Okay, now we will see the domain architecture and glycosylation of an antibody. So the immunoglobulin fold, every domain, variable or constant, folds into a similar structure of two anti-parallel beta sheet connected by loops stabilized by an internal disulfide bond. This shared fold is a basis of a wider immunoglobulin superfamily, which includes TCRs, MHC molecules, and many CAMs. Glycosylation, antibodies are heavily glycosylated, mainly at the CH2 domain of the Fc region. The glycan pattern influences the effector function, including complement fixation and Fc receptor binding. Got it? Now, because only the variable domain groups, not the whole fold, need to differ between antibodies, the immune system can generate enormous diversity while keeping a single stable overall architecture. Okay. Even in that variable region, there are small regions called the complementarity determining region. There are three complementarity determining regions, CDR1, 2, and 3, and those, the one that binds to the epitope of the antigen, only that needs to undergo a lot of genetic mutation to give specificity of specificity to that specific antibody for a particular antigen. Okay, and that is why it is so easy to make such diverse groups of antibody so fast. The hinge region and antigen antibody interactions. The flexible hinge region lets two antigen binding arms swing independently so as a single antibody can bind to two epitopes at varying angles. So let's say these are the two arms of this particular antibody. This can bind to one epitope, this can bind to one epitope. That is why it is called divalent. Okay. Now, antibodies that lack a hinge, for example, IgM and IgE, have reduced arm flexibility because each antibody is bivalent. More antigens are multivalent. Therefore, antigen antibody can cross-link and it drives visible precipitation in classic immunological assays. So, precipitation reaction is one where if you are, let's say, keeping one of them constant, either antigen or antibody, and increasing the other, what happens is there is a bell-shaped curve. That is, initially the binding will increase and then finally they will get saturated, and then it will decrease because the antigen antibody complex precipitates. Okay, it precipitates. It just becomes insoluble after antigen antibody binding. A full big complex is formed and it precipitates. That is a precipitation reaction. Now, what happens when you subject this antigen and antibody to proteolytic enzymes? For example, pepsin and papain. Okay. So, and also beta mercaptoethanol, because there are so many disulfide linkages. So, if you subject it to beta mercaptoethanol, it would be easy for you to even think that all these disulfide linkages are going to break. And then if you run it on an SDS page, you will see only two bands. One corresponds to two heavy chains, the other one corresponds to the two light chains. Okay, and it will completely lose its activity. Now, if you use papain digestion, it is going to cleave above this hinge region. Then what happens is the two FAB regions, they are separated out, but they are still active. Okay, and the FC region is also separated out, but its structure is intact, and so it will retain its effector activity. Okay, so you will see two bands once again. Two corresponds to one FAB, and one corresponds to two FAB, and the other band corresponds to this FC region. Now, if you use pepsin, it will cleave below the hinge region. So what happens is the disulfide bonds are no more there to hold the fragment together. So this kind of degrades. The FC fragment degrades, and the FAB regions, they are still divalent because two different antigens can bind to them. Okay. Now, proteolytic fragments, historic enzyme digestion experiments revealed antibody structure before recombinant technology existed. The cleavage sites are highly conserved across species. So, papain digestion cuts above the hinge, produces two Fab fragments, fragment antigen binding monovalent, plus one is FC fragment. Function split, so FC carries the effector function. Fab retains antigen binding but no effector activity. Okay. In case of pepsin digestion, so pepsin is an enzyme that is there in our body. It is acting on your proteins and peptides in the body, right? So this is an enzyme. Pepsin is an enzyme which looks at specific sequence and cleaves the peptide at that specific sequence, right? So now you see that it cuts below the hinge on the C terminal of the disulfide, producing one this full FAB fragment, and it is divalent disulfide linked pair, and FC is destroyed. It is very small and it is non-functional peptide fragment. It is degraded. Okay, so now we have two MCQs based on this, and we are almost at the end of our course. Sorry, at the end of our today's class. Right, so we are almost at the end of today's class. Now let's see. So papain digestion of an IgG molecule yields one FAB2 fragments and degraded FC peptides, two FAB fragments and one intact FC fragment, two FC fragments and one FAB fragment, only light chain and heavy chains. Pepsin cleaves an antibody on the carboxy terminal side of the hinge disulfide bonds, producing two separate Fab fragments, one single divalent FAB fragment and degraded FC, one intact FC fragment with two Fab arms and only the CH1 domain. So the answer would be B for both. So papain obviously cleaves above the hinge, generating two separate fragments. Both are monovalent. Okay, and the FC region is also intact. Now, this is distinct from pepsin, which cleaves below the hinge. And so what happens? The entire FAB divalent one is there, but the FC portion is degraded. Okay, that is the difference. Now, we have 5 different types of antibodies that we already know. There is IgM, which is first antibody in the primary response. There is no hinge and 4 constant domains are there, creates a pentamer. Okay, the most avid isotype. Now, what is avidity? Avidity and affinity are two different phenomena. Okay, we are going to talk about avidity in the next slide. So see, the next one is IgD. So IgD is co-expressed with IgM as a naive B cell surface receptor, and this is present in very low concentration in the serum, okay, blood serum. And IgG, it is the most abundant in serum, dominant isotype of secondary responses, and only isotype that crosses the placenta. Remember, this is very important. Okay, this is the only isotype that crosses the placenta and moves from the mother into the fetus. Okay, next we have IgA. IgA is dimer. This is secreted in tears, in saliva, in breast milk, and that is why breast milk is so important for the child, because a lot of immunity of the child is developed through the breast milk consumption, right? Now we have IgE. So this mediates allergic responses and anti-parasite immunity. No hinges there. It binds with high affinity with the FC region on the mast cell and basophils. So this is how all the 5 different types of antibodies look like. Okay. Now we have MCQ. Which immunoglobulin type is secreted as a pentamer stabilized by J chain, giving it high avidity despite relatively low individual site affinity? So, avidity means that even if the individual affinity is low, the overall binding affinity is high, and that is called avidity. Okay. Now we have the antigen binding specificity of the antibody is determined primarily by the variability concentrated in which structural elements. Okay, whether it is the entire variable domain framework, whether it is the complementarity determining loops, whether it is the CH1 constant domain, and whether it is the FC glycosylation pattern. So we will look at the right answer. The right answer is IgM. That is, IgM is the first antibody produced in the primary immune response and is secreted as a pentamer linked by a J chain giving it 10 antigen binding sites, right? So there are 10 antigen binding sites in IgM, and that is why even though single antigen binding site has a less affinity, the overall affinity of antigen binding is much more, that is its avidity is very, very high, right? Overall avidity is very high. IgA can form dimers, but the pentameric J chain stabilized structure is characterized by IgM. Now see the next one is complementarity determining regions. While the variable domain has a conserved B sheet framework common to all antibodies, only the exposed hypervariable loops, the complementary determining regions, directly contact the antigen's epitope and account for antigen binding diversity. Now another very interesting thing that I have told you during the class was that there are these nanobodies. So nanobodies are even smaller sections of this antibody, which if you mutate, so a lot of nanobody engineering is going on so that you can change the specificity of this particular antibody to bind to its antigen. Okay, so people are targeting even smaller sections of this CDR so that only that can be mutated. A lot of genetic mutations can be introduced into it so that you can make it bind with higher specificity and higher affinity of interaction with this antigen. Okay. Now, affinity, avidity, and antigen binding loops. Affinity is the strength of a single antigen antibody interaction that is described by association constant Ka. So, this is like equilibrium constant, where the equilibrium constant, as I told you in the class, was that the concentration of the product by concentration of the reactants at equilibrium, right? And in this case, it is concentration of the bound products divided by unbound products, that is Ka. Now avidity, the overall binding strength of an antibody for a multivalent antigen, taking into account antibody valency, two or more binding sites per antibody. Okay, the overall a low affinity antibody with high valency. Valency means how many epitopes it can bind. For example, pentameric IgM can still achieve high overall avidity, which is important early in a primary response. Now CDR loops. So within the variable domain, most residues from a form a structural beta sheet framework. Only a small set of exposed hypervariable loops, that is complementarity determining regions, CDRs, at the tip actually contacts the antigen's epitope. This is why the whole variable domain need not differ between the antibodies. Diversity is concentrated in just these loops, especially the CDR3. Now this is the last MCQ of the day. Which statement correctly distinguishes affinity from avidity in the antigen-antibody interactions? Affinity depends on antibody valency. Avidity depends on single binding site. Affinity is the strength of one binding site interaction, that is Ka or association constant. And avidity is the overall strength accounting for the multiple binding sites. Affinity and avidity are interchangeable terms. Avidity only applies to IgG. Affinity only applies to IgM. Avidity, okay, so that's it. And then which antibody isotype is unique in being able to cross the human placenta, right? So we'll look at the right answers. So affinity refers to the intrinsic strength of a single antigen binding site epitope interaction quantified by association constant Ka. Avidity is the cumulative binding strength of a multivalent antibody such as pentameric IgM or bivalent IgG for a multivalent antigen, and can be high even when the individual site affinity is modest. Which antibody isotype is unique in being able to cross the human placenta? It is IgG, right? This is the only one that can cross the human placenta. That's it for today's class. Thank you. And these are the references that you can go through. Okay, so see you in see you in next Monday's class once again. Thank you, and I'll be ending the call.